Hi everyone. How are you doing? I hope you are doing great. I hope you're doing fine. Today's lecture is straightforward. We'll be discussing the concept about the control of microbial growth, and basically, in specifically, we are going to talk about therapeutics, the therapeutic side of it. Next week we'll be talking about a different aspect of it. So this is today's outline, and then the outline. We'll first define natural and semi-synthetic and synthetic antibiotic drugs. Please let me check and see if my mic is on. Okay, yeah, my mic is on. Mm. And then we will define um selectivity. We will define selective toxicity, and we will also define therapeutic index and identify whether a high or low dose index is preferred in a drug. We will state and define the three categories of drug side effect. And then we will define superinfection and summarize how it develops in a patient. We will define broad spectrum and narrow spectrum antimicrobials. And then we will name the five major cellular target of antibiotics. We will discuss the mechanism of action of the antibiotics on the target and list the major types of antibiotic associated with each target. We will then state the major mode of action of antiviral drugs. We will list the major challenges in eukaryotic antimicrobial therapy. We will state the ways that microbes acquire antimicrobial resistance genetically, and then we will identify the five cellular mechanisms that microbes use to resist antimicrobials. We name and then summarize each method for testing antimicrobial susceptibility. And lastly, we state the ways humans can contribute to antibiotic resistance. So this will be the short outline. We will talk first about the principles of antimicrobial drugs, and we will talk about antibiotics in bacteria, and we also talk of antiviral drugs, and we talk of antifungal, antiprotozoal, antihemolytic drugs, and then lastly we will talk of antibiotic resistance and drug development. Um, So the whole goal is that these microbes are infecting us or attacking us, and we are coming up with a drug or ways to defend ourselves. And these ways have to be selective toxicity. Basically, it means the drug we are going to use have to kill only the microbe, but not the host or the one infected. However, the drugs which are out there, it's not that perfect. So there's advantage and disadvantage of each drug. And in order to fully administer a drug or bring a drug out, you have to also know about the therapeutic index, basically, which measures the ratio of toxicity of this drug. And you calculate this ratio by um, having the dose of the drug that is toxic to the humans and dividing it by the minimum therapeutic effect or the therapeutic dose it can have. So the goal is that when you have a smaller ratio, it means you have a greater potential for toxicity. And when you have a larger ratio, it means the drug is good. So an example is being shown below. Um, okay, let me try and find a pointer. Okay, so an example is seen below here where we have the dose of drug that is toxic to humans is about 50 milligram per ml. And then we have the minimum effective dose, which is 5 ml. It gives about 10. And then we have another drug which is going to have the dose which is 5 milligram, which is dose of toxicity to humans, and the minimum effective dose. That is how much effective dose can you have to be healed, and that is 5. So this is actually bad because the same dose that will allow you to heal is enough to be toxic to your body compared to this one which is having a small, so this is which is having a dose which is smaller. So this dose is actually smaller to make you healthy, but if you take more than that, or if you take around 50 milligram per ml, then it becomes toxic to you. So in order to administer these drugs or bring these drugs out, we have to know about the therapeutic index, which will give us an idea of how many doses is needed in a patient. So, so to talk about this, there are categories of major drug side effect. The first one is direct damage to tissues through toxicity, and we have allergies, and we have some that disrupt in the balance of normal microbial biota. So I'll take it one after the other. So let's discuss about the drug toxicity. The first, so drug toxicity basically can cause a wide variety of side effects. Some can happen in the liver, the kidney, your gut, your heart, and even your lungs. An example of this is overdosing or taking a lot of tetracycline antibody, which will result in tooth discoloring, as we can see from this image over here. And it can also lead to liver damage in pregnant women. In some cases, this drug toxicity can actually result in a gut upset, which results in diarrhea. And most common complaints with oral antibiotic therapy. So most oral antibiotic therapy, when you overdose it, results in diarrhea or having a stomach upset. So now let's move to allergic responsive with drug. So allergic responsive with drug mostly come from the response of the drug molecule or the substance from the body's metallic alteration, body's metabolic alteration of the drug. And we mostly see this with outside as rashes or hives on the person's skin, and they'll be scratching themselves, and can also sometimes include shortness of breath, swelling of lips, face, or tongue, and sometimes the worst extreme cases you will have to faint. And the third part is of toxicity actually results from alteration of the microbiome or the microbiota. So there is a broad spectrum of antimicrobial drugs that treat infections, but will end up destroying your normal microbiota. So you know that every human being there are microbiome in your gut, and so imagine that you are sick, you're taking an antibiotic to go and get rid of some of the infected pathogens or infected microbes in your gut, and then the drug end up killing your healthy microbiome that you have, and also some of the gut microbiome. And what results is what we call super infection. Basically, the pathogen itself end up surviving, and your gut microbiome actually dies. So super infection happens when um, super infection happen when the targeted antibiotics to kill certain microbes end up killing your gut microbe, and that microbe and the targeted infectious microbe end up growing a lot and doubling. So this is a practice. We can talk more about it on Friday. Please practice by yourself. So then let's move to talk about antibiotics, which is targeting bacteria. So antibiotics drug basically targets bacteria, and we have two sources. The first one is natural antibiotics, which are produced by bacteria and fungi, and they are competing between microbes trying to kill each other for food and for environment. Antibiotics were discovered by Alexander Fleming, and what he did was to discover penicillin around 1928, and basically he just saw penicillin. Um, he saw that the penicillin species were growing on plates of Staphylococcus aureus, and basically the penicillin species were kind of producing something that is killing the Staphylococcus. And then that's when we discovered penicillin. That's when he realized that what the penicillin species were giving out or secreting out to kill the Staphylococcus was actually the penicillin drug we have today. And then during World War II, there's an uprise in research and development on penicillin, and people started using it a lot. So, what are the sources of antimicrobial drugs nowadays? So, we have the semi synthetic one, which is basically created by chemically altering the structure of natural antibiotics. And one of this is this turning in of the natural penicillin G to ampicillin, basically by altering this peptide here. And then the next next category is synthetic. Basically, you create it in the lab, mimicking the natural action of natural compounds that we know. And an example of it is silagicin, and basically it mimics the natural activities of penicillin. So the antibiotics actually have a wide spectrum. You can have antibiotics which are having narrow spectrum, which basically means they have the ability to target only one specific group. And then we have ones which is a broad spectrum antibiotics, which means they can target or affect against more than one group of bacteria. So as you can see from here, if you take an antibiotic like isoniazid, it can only target microbacteria, which is responsible for tuberculosis. But if you take something like tetracycline, I don't know if this can show. Okay, pointer here. So if you take something like tetracycline can um, affect get rid of a lot of microbes, including gram-negative bacteria, gram-positive, and chlamydia. Compared to this one, isoniazid, which can only target tuberculosis causing bacteria. And the same thing goes for all of the other ones. So now you appreciate how complex some antibiotics are. Can target more than one group, some can target only one group. So there are five primary modes of action of antibiotics on a bacterial cell. They can either attack the cell wall by inhibiting cell wall repair and synthesis, or by attacking the cell membrane, causing loss of selective permeability. They can attack the bacterial cells' DNA and RNA replication mechanism. They can also block folic acid synthesis in the cytoplasm. And lastly, they can prevent protein synthesis by inhibiting the action of ribonucleases. So first, we will talk about the action of antimicrobial antibiotics on the cell wall of bacteria. So the cell wall of bacteria contains beta lactams, and basically the goal of these antibiotics is to act on the beta lactams and just break them down, basically damaging the cell of the bacteria. And one of such antibiotics that have these roles, some of the antibiotics that have this ability are penicillin, cephalosporins, and then carbapenems. So how does penicillin work? So the mode of action of penicillin basically is to attack the cell wall, basically the synthesis of peptidoglycans in the bacterial cell wall. So penicillin attacks it and prevents the repair and the synthesis of it. They do this by catalyzing the cross-linking of the inhibiting the catalyzing of the cross-linking of the peptidoglycans in the cell wall. And some example of this include amoxicillin, methicillin, and ampicillin. And then there are also some natural and semi-synthetic derivatives that are also being used to basically target this. But the problem is that some bacteria produces penicillinases. Basically, these are enzymes that destroy penicillin. So they are produced by the bacteria and it destroys the penicillin antibiotics, which results in the bacteria being resistant to them. And then, for example, we can have we can combine this with clavulanic acid, which inhibit the beta lactamases. Basically, these enzymes produced by the bacteria, when you combine your penicillin with clavulanic acid, it basically inhibit the activity of this enzyme produced by the bacteria. Then you can then finally kill the bacteria. So next class of drug or antibiotic is cephalosporins, and um, the mode of action is to also target the peptidoglycan synthesis and repair. It was isolated first in 1940s from fungus, and then what it does is it's just basically alters the ring of the so it basically alters the ring of the penicillinase. Penicillinase is basically the enzyme that target penicillin, and then make it possible for penicillin to work. So we have some natural and semi-synthetic derivatives, and and basically some example are the ceftriaxone and then cefdinir, and it keeps going on and on. And the last group is carbapenem carbapenems, and then basically the mode of action is to also target the same cell wall peptidoglycan synthesis and repair. We have natural, we have semi-synthetic, and then the synthetic form of these antibiotics. Originally, they were discovered from streptomycin, and then this is basically more of a last resort antibiotic due to its high toxicity and often taken through IV, and is reserved when other drugs are not actually more effective. And it has been shown to some bacteria have been shown to be resistant strains which are appearing to this drug, and these bacterial strains actually also have the beta lactamase, basically the enzyme that targets this class of antibiotics. And some example of these antibiotics is doropamine, imipamine, and aztreonam. So now we also have to talk about what about the antibiotics that do not act on non-beta that do not act on beta lactams. So we call these classes non-beta lactams. They still target the cell wall synthesis repair, but they do this using a different mechanism. And the first class that we are going to talk about is bacitracin. So these are just a narrow spectrum for superficial skin infections by streptococci or Staphylococcus, and then their mechanism is just to inhibit the peptidoglycan subunit transport. So this time around, they are not inhibiting the peptidoglycan cross-linking itself. They are just inhibiting the subunit transport. The second one is vancomycin. And basically, it also have a same narrow spectrum, which is used for serious infection with drug-resistant staphylococcus. And basically, it binds to growing peptide chains or the peptide bridge and block further synthesis. So now that we are finished talking about class of antibiotics that target the cell wall to inhibit repair, we will now move forward to talk about class of antibiotics that target this protein synthesis, that inhibit protein synthesis. The first class is aminoglycosides. Their mode of action is to inhibit ribosomal function and translation of protein synthesis. Basically, they bind to 30S subunit, leading to misreading of mRNA, and then they end up producing abnormal proteins, which are mostly not useful for the bacteria. These are a broad range spectrum of antibiotics. It means they can target more than one bacteria. They are product of actinomycetes. So these are bacteria in which we got that from. Basically, these are filamentous bacteria. This class of antibiotics comes in both natural and semi-synthetic. Examples include neomycin, which are more common in the lab, and some of them include kanamycin and gentamicin. So the next one is tetracycline or tetracyclines. Their mode of action is to inhibit the function and translation of protein synthesis. Basically, they block the attachment of the tRNA on the A receptor. So as you can see, under normal circumstance, the transfer RNA will come and bind to the A receptor, and then it will recruit the amino acid, and it will move to the next one, and finally to the exit and go out. When you have tetracycline basically comes over here and blocks, preventing the landing of the transfer RNA. And this class of antibiotics also targets a broad range of bacteria. And we have the natural and the semi-synthetic form. Example of these include tetracycline, doxycycline, and this doxycycline actually causes less of tube discoloring compared to the other counterpart. And the third class of antibiotics that target protein synthesis is oxazolidinoids. And the mode of action is basically the same thing, inhibits function and translation, protein translation. And basically, it works by inhibiting inhibiting the initiation of protein synthesis by blocking the P site. So basically, down here, this antibiotic comes in and blocks the P site. So even if the transfer RNA comes here, it blocks the P site, preventing the binding of the 30S to the 50S. Finally, there's no rid of mRNA at all. And then this antibiotics treats it's responsible for treating antibiotic resistant in organisms causing severe infection. So to keep more in our arsenal against drug resistant microbes. The fourth one is pleuromucilins. And basically the mode of action is also the same, prevents protein synthesis. It does this by blocking peptidal trans it blocks it by sorry, it works by blocking peptidotransferases. And basically, if you have it here, it comes over here and just blocks peptidotransferases of the on the 50S, and then leading to just the blocking of bonds, or basically it prevents the transfer of this um, peptide group to the next group. You remember when we discussed it, this peptide group here have to bind to the next available peptide group. When you have this antibiotic, it comes here and just block the binding of the two peptide group, leading to no formation of protein bonds, and no formation of amino acid bonds, leading to no formation of protein. And we have both the natural and the semi-synthetic form. Examples include lefamulin and then retapamulin. Forgive me, these names are really huge. You guys will be hearing a lot of it. So the next group is macrolides, and then their mode of action is also the same: inhibit ribosome function and translation of protein. It does this by blocking protein synthesis, by blocking the translocation. So as we've discussed earlier on, um, once the transfer RNAs give out the amino acid it came with, it have to move here, and then it will now move to the P side. So basically, the movement from the P side to the E side is blocked by this class of antibiotics, preventing the exit and basically blocking and preventing the translocation of the mRNA and blocking everything. And then, sorry, I think I misread it. So the mRNA, I misinterpreted it. The mRNA have to move in this direction, and the antibiotic comes in here blocking the movement of the mRNA itself. So it blocks the movement of the mRNA itself. So it wouldn't move, it's just stuck. So if it's stuck, then it means there's no more reading coming through. And this is actually contains a unique microlate ring, which is responsible for that. And this is also a broad spectrum and type of class of antibiotics. It's actually low toxic, and it's natural, and it's also, we have the semi-synthetic form. We have both the natural and semi-synthetic form of this. Example include azithromycin, and this is more common when you have problems that needs it. So the next one we talk about is class of antibiotics that target folic acid synthesis in the cytoplasm. So it's basically simple. This is the folic acid metabolism pathway, and then anti you have the antibiotics, which are either targeting some in the upstream or downstream or any other place. It just basically targets it, blocking the synthesis of folic acid. And then one of the first classes, the one of these classes, sulfonamides or sulfur drugs. The mode of action is just inhibit enzymes involved in folic acid synthesis. So as you can see from here, it's just basically inhibiting the synthesis, the activities of the PABA, the basically inhibiting the activities of this enzyme, which is dehydropermetrone synthase. So all of these drugs are actually all synthetic. They have a broad range. It means they can target a lot of microbes, a lot of microorganisms or bacteria, a lot of bacteria, basically, not microorganisms. They can target a broad range of bacteria. Example include this sulfacetamide for acne. And then we have to now talk about class of antibiotics that target the DNA and RNA synthesis in bacteria. And then they do this by inhibiting the either the initiation or causing a lot of DNA, causing a lot of either the initiation or causing a lot of mutation in the ligand strand, which we already know will cause a lot of problems. So one of the class we want to talk about now is the fluoroquinolones, and the mode of action is to inhibit DNA replication by preventing DNA unwinding. So from here, it's just basically preventing the unwinding and allowing the RNA or DNA polymerase to start the whole job, and basically inhibit DNA gyrose or topoisomerase, which is responsible for opening of this fork or opening or unwinding of this DNA for initiation to occur. And these are actually a broad spectrum of a broad spectrum of antibiotics. It can target a lot of bacteria. It's very highly potent and is easily absorbed from the intestine when you orally take it. But it's in limited use due to rare but serious side effects, including seizures and other brain disturbances. Example include nalic acid and then levofloxacin. And then second class is rifamycin, and basically an altered is basically an altered chemical is basically altered chemically into rifampin. The mode of action is also the same, inhibit transcription and mRNA synthesis. So basically it inhibits mRNA polymerase directly binding, and then it prevents any transcription that may follow. So as you can see, during normal DNA or RNA synthesis, the polymerase have to bind. Basically, what it does is it just blocks the binding of the polymerase, so there's no more further synthesis happening. And this is more of a narrow spectrum type of antibiotic, and it can pass through the cell envelope of many gram-negative bacillus. And next, we have to talk of the different class, which is the class of antibiotics that targets the cell membrane. So we have polymyxins, and then their mode of action is basically to disrupt the membrane by acting as surfactant or detergent and basically disrupt the membrane permeability. We already know the cell membrane is more selective, allowing specific things to enter the cytoplasm or get out from the cell. And then when you have this class of antibiotics on the bacterial cell, it basically disrupts or destroys it. It destroys or breaks down the cell membrane into pieces, allowing more of stuff entering the bacterial cell or any other thing leaving it, leading to cell death. And it's this class of antibiotic is derived from Bacillus polymyxa, and then we have the natural, semi-synthetic, and the synthetic form. It has a narrow spectrum, basically targeting more of a gram-negative infections and binds to the LPS on the cell membrane, and also part of neosporin. But the disadvantage of this class is that mostly results in frequent toxicity to the kidney, which is not good for you. So you don't have to abuse this. Example of this class of antibody is polymyxin B and colistinine. Then we have the second class, which is daptomycin, and the mode of action is basically is to disrupt the membrane, and then it does this by acting as a surfactant and also basically doing the same thing as we've discussed earlier on. And this was discovered from streptomycin species, and this also have a narrow spectrum. Remember, the first one we discussed was targeting gram-negative. This time around, this class is targeting most of gram-positive infection, and it binds directly to the cell membrane. However, in 2024, there have been a new publish of new classes of antibiotics which have been discovered. Basically, they target the LPS or the lipopolysaccharide transport of the bacteria. And it's mostly, and the ones that we've seen so far is mostly against gram-negatives bacteria. And then there's one leading candidate already in clinical trial, which is A. Bumumani, and basically targeting these classes of bacteria. Please try and practice this, and we can also go over them in class. So now we've talked about principle of antimicrobial drugs, and now we've talked about antibiotics that target bacteria. So now it's time for us to talk about antiviral drugs. We have over 90 antiviral drugs for 9 viral diseases. So you can realize that there's a lot of fewer drugs available for all these viral diseases present. Many are specific to mechanism of actions that won't work as broadly, though, but it's not always true. So, what is the three mechanism of actions of these antiviral drugs that we have? First one is preventing of attachment of the virus to the cell membrane, as we've already known from previous class. The virus have to attach to the cell membrane, but and then it goes inject the viral plasmid and viral DNA, and then the whole assembly and replication happens. But basically, the first class that we know is that this antiviral drug prevents the attachment of the virus to the cell membrane. And then we have the second one is also to prevent like the penetration. So sometimes it can attach, but it just prevent the penetration. And then the third one that the second class that we know is that the antiviral drug basically prevent the nucleic acid synthesis of the virus. And the third group that we know is basically to prevent either the assembling or the release of the virus in the cell. You can realize that this whole mechanism is not directly damaging the virus. It's just basically interfering with the virus life cycle, preventing from either attaching synthesis or release. So now let's talk of examples of antiviral drugs that we know and its mode of action. First one is HIV drugs. Basically, it blocks the attachment of the host co-receptors. So these are the host co-receptors CCR5 or CXCR4. And basically, the goal is that so under normal circumstance, these HIV virus is supposed to bind to the it recognizes the antigen and is supposed to bind to this CCRS or this co-receptor. What it does is that these HIV drugs here is it comes in and the blocks this receptor, preventing the virus from attaching to it. And then we also have other class of HIV drug that basically blocks the fusion um, of the virus. An example, so the virus attaches to the surface, it has to fuse into the membrane and then release it. These fusion inhibitors basically prevent the virus from fusing into the membrane of the host. An example, so let's give an example of the drugs that block the fusion is TRI 999. An example of the ones that block the attachment of the receptors is AMD 070. And then we have other ones that basically blocks the reverse transcription of the viral drug. So an example is AZT and 3T3. Basically, when the virus have attached and released, it have to, because it's the virus, it comes with its own reverse transcriptase, which have to initiate reverse transcription. These NRTs basically attaches these drugs, AZT and co, prevent the reverse transcription of the viral drugs. And then under these classes of anti under these classes of antiviral drugs that inhibit nucleic acid synthesis, the first one is to block the action of reverse transcriptase, as I've talked about. The second one is to block integration or inhibit enzyme integrase. So as you can see, once the reverse transcription is done, the viral DNA or the now made viral mRNA have to now get the viral DNA have to now get incorporated into the host genome. And then this is when the second class of um, antiviral drugs that blocks RNA synthesis come in. Basically, it blocks the integration of the viral DNA into the host genome, preventing more downstream gene transcription from happening. And an example of this include TVCA, as we can see from here, and EVTCA, as we can also see from here. And then the last class of antiviral drug that is present is the one that inhibit the viral assembly and release. An example of this is Tamiflu or Tamiflu. Basically, it's more present in we use it mostly in influenza, and it interferes with the release. So after assembly, you still have the burden and everything present, but the Tamiflu will prevent the release of the or the escaping or the release of the virus from the host. So now that we've talked about antiviral drugs, let's talk about antifungi, antiprotozoa, and antihemetic drugs. So eukaryotes present special problems. Most antibiotics do not work, but a few do. So because there's a similarity between microbial eukaryotic cell and human or animal cell, it makes it more capable of harming human tissues, right? So there's a huge degree of diversity among organism groups. Basically, you are targeting broadly that can be more challenging. So you have these microbes that can infect eukaryotic cell and also human and animal cells. So they have a wide range of infection. And these classes include you include protozoa, fungi, and hermits. An example of drugs that are responsible for getting rid of protozoa, fungi, or hermits include nitronidazole drugs such as metronidazole, tinidazole, onidazole, which is used basically um, against these microbes. But the mechanism of action is actually poorly understood. So we usually don't know how it works, but the goal is that they still work. Please try and practice this one too. We'll talk more about it when we meet again. So lastly, let's talk about antibiotic resistance and drug development. So anti drug resistant microbes are basically microbes that tolerate amount of drug that won't ordinarily be inhibitory. So basically, um, the drug is supposed to kill the microbe, but they end up tolerating it, which is not like normal for them to do. And how do they develop this resistance? So there are two modes. We have the intrinsic resistance, which is resistant to antibiotics that they themselves produce. So like we talked about the discovery of penicillin, where the where the bacteria was using penicillin to kill the streptococcus. Basically, where the bacteria was using the penicillin to kill other group of microbes. That's basically how it is. So that bacteria that is producing the penicillin is it itself is resistant to penicillin. So the penicillin being produced by the bacteria wouldn't kill itself, but it will kill any other bacteria in the environment. So that is what we describe as intrinsic resistance. And then we have acquired resistance, which basically is resistant through when you give out a drug to the bacteria. Some of them will die, some will survive. Those who will survive will develop resistance to the drug. So this is actually called acquired because it's acquired through previously exposed drugs to the microbes or through the bacteria. And there are two major mechanisms of acquisition under acquired resistance. So first we have the spontaneous mutation, which basically um, occurs basically through vertical gene transfer. That means one microbe attaching itself to one bacteria attaching itself to the other bacteria, and then transferring their resistance to them. No, I think I'm confusing this. So we'll talk more about it in the next few slides. So there are two major mechanisms. So we have spontaneous mutation and horizontal gene transfer. So spontaneous mutation, basically It's a random change in DNA arising from DNA errors in DNA repair. So as we talked about it earlier on, the leading strand, there's no mistakes happening on the leading strand during DNA repair, and then there's mistakes happening on the lagging strand due to the multiple priming of the RNA primer, leading to Okazaki fragments happening. So these fragments will result in some mutations, which will then be transferred from the bacteria to the daughter cells when it breaks apart, or when binary fission happens, where the bacteria have to make multiple copies of itself. So there is minimal chance that mutation will be advantageous under spontaneous mutation, which is occurring from the lagging strand. And there's actually smaller chance that this mutation will confer to drug resistance. But large microbial populations and constant mutation, there's a large microbial population that have it, leading to constant mutation rate. And these are sometimes beneficial mutations do occur. So one prime example of this include there's antibiotic in here and in this media, and then the original bacteria which was cultured in here is not resistant to the antibiotic and it died, and then the same media, the same antibiotic was used to culture the other bacteria which have now developed resistance through spontaneous fusion, through spontaneous mutation, and now this time around it's able to grow in the media because it have acquired a mutation which is beneficial. So there's also other ways in which um, bacteria can resist this. So one of them is the natural selection of drug resistance. So in any large population of microbes and you want to get rid of them, without the drug without you giving them antimicrobial drugs, you find a drug resistant mutation, which are mostly not many, but they are little. In any microbial community, there's some which are already resistant to drugs. They are not many. But when you treat them with antibiotics, what happens is that if you treat them with antibiotics, what happens is that you end up killing the ones which are not resistant, and you have the ones which are resistant who will end up multiplying and growing more, which basically means um, you are going to have more problems dealing with them in the future. So without drugs, there's less of a problem. More of drugs or more of antibiotic usage will contribute to the spontaneous growing of the resistant ones. This is mostly done under what we call selective pressure, basically getting rid of the non-resistant one and growing more of the resistant ones. So now let's talk about the second mode of antibiotic resistant development in the microbe, which is horizontal gene transfer. So basically, you have bacteria A sending its plasmid over to bacteria B, and then bacteria B now becomes resistant. And then this can be happening through several mechanisms, including conjugation, transformation, and transaction, as we've discussed earlier on. And then this kind of antibiotic resistance is more of frequent and more popular among nature, and it's a major contribution of resistance compared to the one that we first discussed, which is the spontaneous mutation occurring from DNA replication. So, what are drug resistant genes encoding? Right? What are these plasmid encoding, or what are these mutation encoding? This mutation or these plasmid which are passed on from one bacteria to the other bacteria, actually, so these plasmid, let me take it again. So these plasmids that are passed on from one bacteria to the other, or the concerning the spontaneous mutation, these new mutations or these new gotten genes that are either coming from RNA replication mutation, DNA replication mutation, or transfer of plasmid from one bacteria to the other. These new gotten genes or these new genes develop. They actually mostly encoding for a lot of cellular mechanisms. So one of them will include that these new genes or these new mutation will be responsible for synthesizing of new enzymes that will either inactivate the drug, or these genes will be coding for proteins that will allow for permeability or uptake of drug to decrease. So basically, they are shutting down all of the all of the pores or all of the channels that the drug can go through into the cell membrane, into the bacteria, they develop new mechanisms or new proteins to shut those pores down. Thirdly, these genes might be coding for proteins that are responsible for developing the drug pump or basically upregulating the activities of drug pump. So drug gets into the bacteria, it pumps it out immediately. The bacteria pumps it out immediately through the cell membrane. Or these genes might be coding for proteins which are which are responsible for binding to drug sites on target, which will bind to the drug site on target. So where the target in which the drugs are supposed to bind in the bacteria, these proteins will go in there and change either they bind to the binding pocket where the drug will initially go and bind to, and then block it from binding to that specific spot, or they will decrease the affinity or the number of places which are available for the drugs to bind. And lastly, um, some of the genes will basically produce proteins or produce new pathways, result in new pathways. Basically, they will shut the old pathways that the antibiotics are supposed to target, and they will start a new pathway, which they develop a new pathway which the new drugs or the antibiotic drugs do not know and don't know how to interfere. So let's go through them one after the other. So the first one is the synthesizing of new enzymes that will inactivate the drug. Basically, this results in the breakdown of the drug itself. One example is the beta lactamases, which are produced by um, penicillin producing bacteria, penicillin producing bacteria. Basically, this mode of antibiotic resistant development happens through horizontal gene transfer. And basically here we have the active penicillin, and then they will secrete their penicillins and just degrade or makes this part inactive. Basically, the penicillin is no more active. And then the second way of resistance mechanism is permeability or uptake of drug is decreased. So they will synthesize proteins that will prevent this. So here you can see that they have receptors available, which then the drug will bind to it and it will go and more of it will kind of permeate into the cell membrane and go into the bacteria to work. But as you can see, they've synthesized new proteins that changes the shape of this receptor. So you can see this shape is different from this shape. This time around, when the drug comes in, it doesn't recognize the receptor and it cannot bind to it, basically resulting in resistance to this drug. So it's coming usually from mutations. So imagine this as a puzzle where there is a specific piece that is responsible for fitting to specific space, and then the bacteria just changes the shape of this piece. It means you can't put it back there again. And then there's a different mechanism, which basically is a drug is immediately eliminated through by drug pumps. So normally the bacteria binds, the drug will binds to the receptor and goes into the microbe and then bacteria and then attack it. But basically, what they do is the new proteins or the new mutation they get, they can activate or get new drug pumps. And then basically, once the drug enters, it pumps it out. It enters, it pumps it out, and then it never get to attack the bacteria. And this mode of mechanism is also acquired through horizontal gene transfer. And then the fourth one is drug binding sites on target are decreased in number and affinity. So as you can see, let's say the drug was supposed to bind to this specific area and then inhibit the this specific area on the ribosome and then inhibit protein synthesis. Basically, what the new gene that is acquired or the new mutation does is it changes the spot. As you can see here is triangle. It just changes into a circle. And then when the drug goes in there, it never recognizes the target anymore because it's now a circle like the puzzle piece. And then the bacteria gets to escape this antibiotic inhibition. And this mode of mutation is acquired through spontaneous mutation or horizontal transfer. The last one is the metabolic pathway modification or alternative pathway being used. So basically, what happened is that we have drugs that target these enzymatic pathways and other pathways of the bacteria trying to block it. For example, it can be folic acid metabolism, it can be nucleic acid metabolism. Let's take an example here. If a drug is supposed to block this pathway, basically moving of B to C, and blocks it, and then the bacteria don't produce this product and it will die. What the bacteria does, the new genes or the new mutation it has have can then synthesize a new pathway, basically from A to B instead of C. It changes to C1, then it escapes the drug. So now the drug goes in, it doesn't find any target to actually target. And this is also another mechanism that the bacteria have developed, which makes it resistant to antibiotics. So this give them the ability to kind of like turn off this pathway and then turn on this pathway. So now we've now understand how the antibiotic resistance happen and what is their mechanism. But how do we actually test this in our lab spaces? So to test this antibiotic resistance susceptibility, we have a first test that we want to talk about, which is the Microbial test. And this is basically you have a bacterial lawn, and on it you put antibiotic discs, and then you calculate or you kind of measure the zone of inhibition, comparing it to the standard. So here we have a standard which is an enrofloxacin, and then we know for sure that it's supposed to have the inhibition long around 70 millimeters to 22 millimeters, right? So you already measure it. Then now you come in with all groups of antibiotics, and then see how much of resistance or how much of a microbes these antibiotics can kill. So as you can see, this specific one have a bigger circle around it. This one have a smaller. So this it means this one might have the bacteria might have antibiotic resistance towards this one compared to these ones, and it keeps going on and on. So this table actually represent some known drug resistance from the Capibos test. And then, for example, here gentamicin is known to have a susceptibility rate of 13. And then if when you have a long space of more than 13 millimeters, it means the bacteria are susceptible to it. If you have a long space of less than 12, it means they are resistant to it. And some of the examples of which has been shown is that if you treat gentamicin with staphylococcus, you end up having 16, which means they are more sensitive to gentamicin. The same thing will apply to when you treat these same staphylococcus with vancomycin. They have 15, and over here it means when you have 12 or more, they are sensitive. So it means vancomycin is more detrimental to staphylococcus. However, if you have streptomycin, staphylococcus are actually resistant to streptomycin because you have more of a long space of 11, and here it shows that if you have 11 or less, then it means the streptomycin is resistant. So that goes on, and it applies to all of the other antibiotics. The second test is the E-test. It's similar to the Capibor test, but with the range of antibiotic concentration on a strip. So you realize the first one, we just put the antibiotics in circles on it, but this time around we have all of we have one antibiotic and we have them in a range of different concentrations. So here is the highest and here is the lowest. And as you can see from here, in the highest one, you can see most of the bacteria have been cleared here, which is in the red one. And when you come to the lower part, it's not working on the bacteria, which shows these concentrations, the bacteria are resistant to it. And we have the third one, which is the minimum inhibitory concentration. Basically, just like a serial dilution way, you have one which is a negative control, which is zero, no antibiotic present, and you just keep increasing the concentration of the antibiotics till you get to a concentration where none of the bacteria survive, and then you get to realize that this concentration is actually the best for inhibiting or killing of this bacteria. Remember, this is actually done in a test tube, so you just have a range of test tubes and you do this in it. So as humans, we have a role to play in antimicrobial microbial resistance. And the first role that we play mostly in it is the misuse of antibiotics. About 75% of our antibiotic prescription are for throat, sinus, and upper respiratory infections, which are more like and which are more likely to be misused by humans. So there are tons of excess drug produced in the US and exported to other places. And then most of the time, people end up taking it, even if they have a little slide of condition, they just keep abusing and taking it, which end up in the microbe developing resistance to it. The second place that human play part in it is the hospital environment. So because we are in the hospital, there's a lot of pathogens being exposed to a lot of drugs. You have susceptible patients with weakened defenses, which will end up mean you have one patient, they are always sick, then you have to give them a lot of drugs. And sometimes through horizontal transfer, some of the microbes in the hospital environment also end up transferring some resistant gene to the other microbes, making them also more resistant. And then the third one is that nearly 80% of antibiotic in the US is given to livestock. So you realize that not only humans are using antibiotics, livestock are also being given antibiotics during livestock production. And these livestock, they take these antibiotics and it helps them improve their health. It cures most of their infections for them. However, the deposits of the antibiotics is mostly deposited in the gut microbiome of this bacteria, and then they share this resistant plasmid with each other. At the end of the day, we end up eating these microbes, and sometimes some of these I'm sorry. At the end of the day, we end up eating these animals, and sometimes if we are not lucky, some of the drug resistant bacteria will end up passing their plasmid to other microbes that have the ability to jump on or co-infect both human and the animals, which means they can give us diseases. So what I mean is that you have a pig, you cure the pig with antibiotics, the gut microbe of the pig develop resistance to the antibiotic, and then you sacrifice the pig and you eat the meat. You end up tossing out all of the intestines and other part of it. In nature, these microbes can survive and pass on their plasmid to other microbes that have the ability to infect we humans. So at the end of the day, the pig's microbes are going to transfer their plasmid to the human infecting microbes, and the human infecting bacteria can then come back to infect us again. And the fourth point is global transfer and globalization of food products. So as more food product is being produced, you end up sending some of the product to other countries, which means you might mistakenly send some microbes or some bacteria which have resistance in certain diseases, and they go over there and also transfer plasmid to other bacteria, causing more of a problem to the world. The last problem is that drug development is limited. Actually, it takes about 10 years per drug to 10 years to produce one drug. And then it has to go through a lot of phases like phase one, phase two, phase three of clinical trials. And it costs over $1 billion to produce just one drug. And here is the problem. There's a high failure rate when it comes to drug development. You can just produce it and it doesn't work, so you have to stop and start again. But because it's taking us about 10 years, it means most of the bacteria are going to develop resistance quicker compared to how quick we develop our drugs. And one example is actually being seen here, where the flucloxacillin was identified in 1980, and this flucloxacillin was identified in 1988, and by 1990 they developed the drug and it was approved. And within a few time, the same thing happening here in 2000, and by it was approved, and within few years it became resistant to the drug, and then they have to develop a new drug for caspofungin, which was identified in 2001, and by 2004 it became also resistant. So basically that's it. So you develop the drug in 2001, by 2004 it's already the bacteria is already resistant to it. which now becomes a problem. It means you have to develop another one for it. So there's actually new approaches to antimicrobial therapy. These are alternative approaches we are using to battle this problem. And basically, instead of developing more drugs, we are going to the first one is to use host molecules to enhance immune system. So you have to target the host or the infected person's molecules to enhance their immune system to fight against these viral infections. And also the second part is to also use bacteriophage to attack this bacteria, which is currently being used in when you have a very, very chronic bacterial infection. And the third one is to promote the use of probiotics and prebiotics. And the last one is to use fecal transplant. So what is so so these ones are self-explanatory. I'll talk more of the prebiotics, and fecal transplant. What is probiotic? Probiotic is basically live microbes in food or beverages and supplement. And basically we use them to replace microbes lost during antimicrobial therapy. And it's there for it to boost the host microbiota, which is already present. And then we have prebiotic nutrients. These are nutrients that encourages the growth of the beneficial microbes already in your intestines. And some of these include example is the fructans. Example is fructans, which are used to encourage the growth of your gut bifidobacterium in the large intestines and then discourage the growth of other pathogens. And then we have treatment such as the fecal transplant. Basically, this is mostly used for reoccurring Clostridium difficile infections. And basically, what we do is, as in its name, you just transfer feces containing you transfer feces which are containing beneficial normal biota from a healthy patient, and you transfer it to the affected patient through colonoscopy. I don't think this will be a fun thing for scientists to do. So, please try and practice your case studies on antibiotic resistance, which you get extra credit for, and also practice the dark side of and try and finish the dark side of antibiotics. Sorry, these are not for practice. These are actually something you have to do to get extra credit. Make sure you come on Friday and see you on Friday. Remember, we have TA hours on Mondays and on Wednesdays, both on Zoom. In case you need help, please go to the classroom or attend the Zoom and talk to the TA. In case you have any question, please reach out to me and have a nice afternoon. Okay, bye.